this is going to be the final video of decoding your future. So we are going to finish off strong with biotechnology. Now first, what is biotechnology? Biotechnology is the use of technology and natural biological processes to solve a human problem. An example of this is we use biotechnology to produce vaccines. Now, in biology, we will focus on the use of biotechnology to manipulate an organism's genome, which is known as their genetic information. So, this process is known as genetic engineering. So, today we will research several different aspects of genetic engineering and discover their implications on humans and the world around us. Now, advances in DNA technology. As we have learned, DNA can be manipulated. Scientists use several techniques to manipulate DNA. Since DNA can't be manipulated by hand, scientists use chemicals, computers, and even bacteria to manipulate an organism's DNA. So, the manipulation of DNA, known as genetic engineering, has resulted in advances in medicine, forensics, agriculture. So let's talk about genetic engineering in regards to medical purposes. We use it to produce medicine, such as insulin, modify lab organisms to make them easier to study. We also use it in improving crops. So we make our crops resistant to diseases, pests, frost, herbicides, and so many more things. We improve crops by offering better nutritional value to them. We delay ripening and rotting so fruits and vegetables will last longer on store shelves and in your home. We produce larger fruit. Now, in forensics, we are able to match crime scene DNA to suspect DNA. So, first, let's talk about biotechnology in forensics. DNA fingerprinting. So, one type of genetic engineering is DNA fingerprinting. DNA fingerprinting, or DNA profiling, is a laboratory technique used to identify an organism based on their DNA profile. So, this is how crime scene investigators will match crime scene DNA to suspect DNA. Now, we also have gel electrophoresis. Gel electrophoresis is the process of creating a DNA profile. So we have our agarose gel down here, and we attach it to a machine that helps us build up the lines of the DNA in the gel. And it does that by using an electrical current to separate the DNA fragments into a barcode. So applications of DNA fingerprinting can be used in forensics, paternity, which is determining the father of a child, identifying victims. So for DNA fingerprinting, you have blood, hair, and cheek cell samples that can all be used to create a DNA fingerprint profile. So here we have the blood stain or the sample, and we have samples from Bob, Sue, John, and Lisa. Now we have people over here that is just known as mother, child, male 1, and male 2. So we are going to be focusing on male 1 right here. So we're going to try to see if his DNA will match with any of the DNA over here. And if you notice, John matches the blood stain bar for bar. But if you look over here, it is also relatively close. So, this is how we use DNA fingerprinting. We look for the matching in the barcode to determine if there is any correlation or connection. Now, let's practice with the case of desert bones. All right, let's get into biotechnology in medicine. So, biotechnology in medicine, this is something that scientists have been working hard to learn more about human genes. So, we have the Human Genome Project, which has united scientists in a common goal to figure out the sequence of all the genes within our bodies. They've successfully completed mapping our genes in 2003 by identifying all of the 80,000 genes in human DNA. So the main goal of this project was ultimately to find where certain genes were on chromosomes so that we may correct genetic disorders or eliminate them altogether. So, in medicine, genetic engineering has been used to mass produce insulin, human growth hormones, infertility treatments, antibodies, vaccines, and many other drugs. Now, today, researchers use recombinant DNA technology to analyze genetic changes. So they cut, splice together, and insert modified DNA molecules from different species into bacteria or other types of cells that rapidly replicate and divide. So the cells copy the foreign DNA right along with their own DNA. An example of this is what we use to make insulin. A plasmid is a circular form of DNA in bacteria. Scientists remove the bacterial plasmid, splice it, or cut it open, and insert the desired human gene. The modified plasmid, also known as the recombinant DNA, is then reinserted into the bacterial cell. So the bacterial cell will then produce whatever protein the inserted gene codes for in mass quantities. And this is how insulin is made. Now, when DNA from one organism is inserted into the original DNA of another organism, the new DNA is called recombinant DNA. And these organisms are called transgenic organisms. And finally, let's talk about biotechnology that is used in agriculture. Biotechnology provides farmers with tools that can make production cheaper and more manageable. For example, some biotechnology crops can be engineered to tolerate specific herbicides, which make weed control simpler and more efficient. Other crops have been engineered to be resistant to specific plant diseases and insect pests, which can make pest control more reliable and effective and or can decrease the use of synthetic pesticides. So uses of biotechnology in agriculture is that plant biologists have used DNA technology to produce plants with many desirable traits. Now, these are called GMOs, or genetically modified organisms. So, here's just a few traits in agriculture that you can read through. So, one example of a trait is pest resistance. So, an advantage to this is that there is less damage by the insect, virus, bacteria, or any other things. And something that has this trait is corn. Now, let's look down here. A trait that we also like is delayed ripening. So, this way it can be shipped with less damage. An example that we use this with is tomatoes. And I'll give you guys just a few minutes to read through those. Now, there are pros and cons of genetic engineering. So let's talk about the risk of genetic engineering first. We will see decreased genetic diversity, a risk of unintentional mutations, which will introduce resistance into our genetics, the safety of genetic engineering, and it also brings some ethics into question. And where do we draw the line? So, when we talk about ethical questions, where we draw the line and the safety of others, a big con that is out in the world right now with GMOs is that we have not had genetically modified foods long enough to truly know what the negative sides will be. It hasn't been around long enough for us to see if there will be changes to our DNA or if it will cause mutations in the future. So that brings in the question of if it is truly safe to be introducing it to the masses. Now, some benefits of genetic engineering is that we have introduced new medicines and vaccines, which has helped people live longer. So we have more crops with favorable traits. We've cured genetic diseases as well as solved crimes. So that's something that, have also done really good. So that's where you have to make the choice of, do the pros outweigh the cons for genetic engineering? Is the ability to create medicines and vaccines and solve crimes, yeah, that's great. But is it worth continuing when you bring in the possibility of safety concerns for the population? So, these are just a few things for you to think about in regards to biotechnology. All right, guys, I hope you enjoyed this. I find biotechnology very interesting. And that is going to be the end of Decoding Our Future. Thanks, guys.